Hello everyone, as we already covered prokaryotic cell, here we are going to cover about eukaryotic cell in detail. Coming to the etiology of the eukaryotic cell, it is Greek word where eu means true, karyon means nuclear, where it contains true nucleus and membrane-bound organelles like endoplasmic reticulum, mitochondria, vacuole, lysosomes, Golgi apparatus, etc. It also contains complex locomotory and cytoskeletal structures which helps in locomotion, and these provide strong structural support to the cells. The genetic material is organized into chromosomes where the nucleus is covered by nuclear membrane. Coming to the examples of the eukaryotic cells, these are examples: protista, fungi, plant and animal cells. As we all know, eukaryotic cells are not identical. Let's differentiate between plant cell and animal cell. Here, plant cell contains cell wall as outermost protective layer, which is made up of cellulose. These plant cell surrounds the plasma membrane, where in animal cell the cell wall is absent. The outermost covering is the plasma membrane. Coming to the size of the cell, plant cell is larger than the animal cell. In mature plant cell, it contains large central vacuole which occupies up to 90% volume of the cell. Coming to the plant cell, animal cell, here the vacuoles are small, many and temporary. Coming to the plant cell, here the plastids are present which involved in photosynthesis. The plastids are absent in the animal cell. The centrioles and centrosomes are present in the animal cell and absent in the plant cell. Let's discuss about structure of plasma membrane, which is also known as cell membrane. As we all know, it is a selectively permeable in nature. It regulates the movement of substances in and out and interacts with the outside world. The structure of plasma membrane is studied on the human red blood cell, and detailed structure of the plasma membrane is studied after the invention of electron microscope in 1950s. Coming to the composition of the plasma membrane, it contains lipids, proteins and carbohydrates. Coming to the lipids, these are 40%, proteins which are 52%, and it also contains carbohydrates on the cell membrane which is very less amount. Coming to the lipids, these also contains phospholipids and cholesterol. The phospholipids which has hydrophilic polar end towards the outside the membrane, which contains saturated hydrocarbons, and it also contains non-volatile towards inner side, which is hydrophobic in nature. Coming to the proteins, on ease of extraction, these are different classified into two types: integral proteins and peripheral proteins. Integral proteins which are partially or totally buried in the membrane. These are also known as transmembrane proteins because these proteins which are present throughout the lipid bilayer. Coming to the peripheral proteins which lie on the surface of the plasma membrane. Carbohydrates, as we already discussed, these are present in very less amount. The most improved model of plasma membrane, which is known as fluid mosaic model, this is proposed by scientists Singer and Nicholson in 1972. According to this model, they said that the lateral movements of proteins within the lipid bilayer is due to quasi-fluid nature of lipids, and this fluid nature of lipids which helps in growth of cell division, endocytosis and secretions, etc. And this ability to move within the membrane is measured as its fluidity. These are the various cell organelles that are present in the eukaryotic cell, like nucleus, lysosome, ribosomes, endoplasmic reticulum, mitochondria, Golgi apparatus, etc. Let's discuss structure of each and every cell organelle and functions performed by them in detail. Coming to the endomembrane system, it composed of group of membrane-bound organelles like endoplasmic reticulum, Golgi complex, lysosome, vacuoles, which are structurally different but functions in a coordinator manner. The mitochondria, chloroplast which is present in plants, and peroxisomes, these are not the components of endomembrane system, as these are not functions in a coordinated manner. Coming to the endoplasmic reticulum, these are the tubular structures which are scattered in the cytoplasm. Let's discuss the structure of endoplasmic reticulum in detail with the help of diagram. Here, the endoplasmic reticulum, it divides its intercellular space into two compartments, which are luminal compartment, which is present inside the endoplasmic reticulum, and extraluminal compartment, which is present towards the cytoplasm. Again, based on presence of ribosomes, these endoplasmic reticulum is divides into two types. Those that pierce ribosomes on its surface are known as rough endoplasmic reticulum, which involved in the synthesis of proteins, and these rough endoplasmic reticulum, which continues with the outer membrane of nucleus. Coming to the smooth endoplasmic reticulum, these do not contain any ribosomes on its surface, and these involved in the synthesis of lipids, and these also synthesized lipid-like steroid hormones. Let's discuss about Golgi apparatus in detail. These are densely stained reticular structures which are present near the nucleus of the cell. These are first observed by Camillo Golgi in 1898. Coming to the structure of the Golgi apparatus, it consists of four parts, which are cisternae, which has scissor-forming phase, and tubules, vesicles, and Golgian vacuoles. Let's discuss structure of each part in detail. Coming to the cisternae, these are flattened sac-like structures which has scissor forming phase and maturing phase. As these two phases different to each other, these are interconnected in its function. These cisternae are parallel to each other and arranged concentrically near the nucleus with diameter 0.5 micrometers to 1 micrometer. Coming to the tubules, these are flattened round structures which are arises at the periphery of the cisternae. Vesicles, these are small round structures which are arises at the edges of the cisternae. Coming to the Golgian vacuoles, these are large spherical structures of the Golgi apparatus. Let's discuss functions of the Golgi apparatus. These helps in processing, packaging, and transporting the materials to the intracellular targets or outside the cell. The proteins which are synthesized in the endoplasmic reticulum are transported towards the cisternae of the Golgi apparatus and fused to the cis phase of the Golgi apparatus and moves towards trans phase and maturing phase of the Golgi apparatus, where these proteins are modified before they are releasing out of the trans phase, and these are later acts as lysosomes and perform various functions in the cell. Let's discuss the structure of lysosome in detail. These are tiny spherical sac-like membrane-bound vesicular structures which are formed by packing in the Golgi apparatus. These are rich in hydrolytic enzymes like hydrolysis, which are lipases, carbohydrates, proteases, and nucleases. These are active at acidic pH and helps in capable of digesting carbohydrates, proteins, lipids, and nucleic acids. This also contains transport proteins on the surface of them. Let's discuss about mitochondria. As we all know, this is cylindrical, a sausage-shaped double membrane organelle which is present in the cytoplasm of the cell, which has 0.2 to 1 micrometer in diameter and on an average, which is 0.5 micrometers, coming to the length of the mitochondria, which is 1 to 4.1 micrometer in length. And to see the mitochondria under the microscope, we have to use special stain known as Jonas green B. There are variable number of mitochondria are present in the cell, different types of the cells depending on the physiological activity, shape, size and energy requirements of the cell. Let's discuss structure of mitochondria in detail with the help of diagram. Coming to the structure, it is double membrane bound organelle which contains two membranes, that is outer membrane which continues as limiting boundary of the mitochondria and inner membrane which forms number of infoldings known as cristae, and the lumen of mitochondria again divides into two compartments. Outer compartment which is present between the outer and inner membrane of the mitochondria, which is known as intermembrane space or perimitochondrial space. Coming to the inner compartment which contains dense homogeneous substance known as matrix. These contains circular double-stranded DNA molecules, RNA molecules, ribosomes which are 70S, and these also contains components required for the protein synthesis. Coming to the functions of mitochondria, these are the sites of aerobic respiration and these are also known as powerhouses of the cells, as these contains enzymes required for the synthesis of ATP and these produces energy required by the cell to perform various functions. There are two membranes in the mitochondria. The two membranes have its own specific enzymes which are associated with the mitochondrial function. Let's discuss about ribosomes. These are observed by George Pellet in 1953 as dense particles. These are non-membrane bound organelles which are present both in prokaryotic and eukaryotic cell as 70S and 80S ribosomes. It has two subunits, larger subunit and smaller subunit, which is 60S and 40S in the eukaryotic cell. Here in prokaryotic cell 50S and 30S, this is the larger subunit and this is the smaller subunit. Here S stands for Svedberg unit, which is segmentation coefficient. Here it measures the segmentation rate. Coming to the structure of the ribosomes and its functions, the ribosomes which helps in synthesis of proteins and translating mRNA into specific sequences of amino acids. Let's discuss about cytoskeleton. These are elaborate network of filamentous proteinaceous structures like microtubules, microfilaments and intermediate filaments which are found in the cytoplasm of the cell and helps in the providing strong structural support to the cell movement of the cell and maintains the shape of the cell. Let's discuss with the help of diagram. These are microtubules which are hollow cylindrical structures found in the cytoplasm of the cell and made up of tubulin protein and which forms the cytoskeleton of cilia and flagella. These are microfilaments. These are unbranched structures found in the cytoplasm of the cell and composed of actin and myosin proteins, helps in the cell division by forming an furrow. These are intermediate filaments. These are hollow filamentous structures helps in the forming scaffolds for chromatin material. Let's discuss about structure of centrosomes and centrioles. Here the centrosome consists of two cylindrical structures which lie perpendicular to each other and has an cartwheel-like organization are known as centrioles. These are surrounded by amorphous pericentriolar materials. Let's discuss structure of centriole in detail. Coming to the structure of centriole, it has 9 evenly spaced peripheral fibrils which are known as triplet fibrils made up of tubulin protein, and these triplets are linked with adjacent triplets with the help of AC linkers. and it also has central proteinaceous hub which are connected to these triplets with the help of radial spokes. These centrioles forms the basal body of cilia and flagella, and spindle fibers gives rise to spindle apparatus during the cell division. Let's discuss cilia and flagella. These are hair-like outgrowths of cell membrane. Coming to the cilia, these are small structures which helps in the movement of cell and surrounding fluid. Here, the flagella, which is longer one, which is responsible for cell movement. Structurally, the prokaryotic flagella is different from the eukaryotic flagella. Here, it consists of basal body, rootlets, basal plate, shaft or axoneme. Let's discuss it with the help of diagram. Let's see the structure of flagella. It's an axoneme, a shaft of the flagella. It contains 9 microtubules with 2 central tubules which are connected with bridges and enclosed by central sheath. So these flagella has an arrangement of 9 plus 2, where 9 means it has 9 microtubule doublets and 2 means 2 central tubules. And both cilia and flagella emerge from the centriole-like structures called basal bodies.